Today we will cover gluconeogenesis. I will talk about the organs where gluconeogenesis can be performed, the starting materials, the pathway itself with the key enzymes, and finally the regulation of gluconeogenesis. Before we go further, again, it's just a reminder that the Word file for this lecture is posted on Entrada under the basic medical metabolic pathways folder. So if you want to read rather than listen, please read the material over there. Also, I found a couple of recordings on YouTube that might be useful for you as an overview. So there is a short gluconeogenesis overview. There is another one which actually introduces some steps for you. And this is the same folder as I told you in the glycolysis material, which compares and contrasts the regulation of glycolysis and gluconeogenesis. So I wanted to start with a definition. So gluconeogenesis means that glucose new synthesis from scratch. So it doesn't mean that there is glycogen where the glucose is already there and we just liberate the glucose from that. We put together the glucose from small molecular weight material. Now, why do we need gluconeogenesis? Think about these particular cells. These cells are all glucose users. Testis, cornea, lens, medulla, kidney medulla, red blood cells are anaerobic. So the only food for them is glucose. The nervous cells of the nervous system are aerobic, but you may recall that the blood-brain barrier does not let through fatty acids, which means that a lot of glucose will be used by the cells of the central nervous system. Which organ is capable of doing gluconeogenesis? And I put there 90% and 10%. What this means is that the major organ that performs gluconeogenesis is liver. But when we starve, then the kidney can help out and also produce some glucose for us. Now, which hormone signals to start gluconeogenesis? Which hormone signals to stop? And what is the signal for hormone secretion? So let's start from backwards. So what is happening? Why do we need gluconeogenesis and when? We need it to continuously feed cells that rely on glucose. When do we need to put out glucose from the liver? When the circulating glucose level in the serum is dropping. This lower than normal glucose level signals to release glucagon, and also low glucose and starvation fasting is a signal, as a stress signal, so epinephrine will help too. But main hormone is glucagon. What is opposing glucagon? Insulin. So when the glucose level, circulating glucose level is high, insulin will be secreted by the pancreas. It stops gluconeogenesis and gives the signal for glycolysis to start. I really like this figure because you actually have glycolysis, which are the black arrows, and gluconeogenesis, which are the red arrows, depicted on the same figure. It also contains two of the very important enzymes of glycolysis. I don't know why the name of the third enzyme is not here, which is hexokinase or glucokinase. And you may remember from the previous lecture that when you see this curved arrows, it means that these are the enzymes that are irreversible in the glycolytic process, which means that these enzymes need to be circumvented by other enzymes to turn back glycolysis and actually produce glucose. This figure also says that it is in the liver and glucagon is the signal, so good to remember. And it also tells you what simpler molecules can be used to convert them to glucose. So let's start from the most important uh, uh, basic material, which is lactate. Lactate is produced by lactate producing cells, the anaerobic cells, all the time. They come to the kidney, to the liver, and right away they can be used to produce glucose. The second most important starting material are amino acids. Amino acid carbon skeleton can give you material to be turned to pyruvate or oxaloacetate, and in these 2 points start gluconeogenesis. Now, very important thing to remember when we start from amino acids have the amino group which is liberated from the carbon skeleton in the form of ammonia. Ammonia is toxic. Ammonia needs to be handled. And in a later lecture, I will talk about how we remove this ammonia and that we channel in situ there in the liver cytosol to the urea cycle to turn this very poisonous material of ammonia to urea, which we can excrete easily through the urine. Now, one more thing that, and I will revisit this later, that there are molecules that can produce oxaloacetate and directly, and then there are molecules, the odd carbon fatty acids, that also can be turned into oxaloacetate. And the other important thing to remember, because now we start building how the different pathways within one cell can support each other, that when gluconeogenesis is running, we need an energy source. And the energy source is nothing else but fatty acids that go to the mitochondria and produce energy. But fatty acids come to the liver and can be liberated from triglycerides. And when triglycerides are broken down to fatty acids, and glycerol also needs to be transported to the liver, and glycerol can be also turned to glucose. So when you review for this material, I think this is a great graph to use to remember a lot of things. So this is what I meant before. What is the relationship? Glycolysis is running to other pathways. Glycolysis is running in other cells. Lactate is produced, goes to gluconeogenesis. Fatty acids and triglyceride metabolism can also support gluconeogenesis through glycerol coming from triglycerides and propionyl coenzyme A coming from odd carbon fatty acids. Please remember, acetyl coenzyme A, which is produced from fatty acids, cannot be turned back to produce glucose. However, when you handle odd carbon fatty acids, after the last round of beta oxidation, a 3-carbon unit will be produced. One single 3-carbon unit, all the others will be acetyl coenzyme A. And then it can be turned into oxaloacetate, and oxaloacetate can go turn back to phosphoenol pyruvate, and then it can produce glucose. Of course, amino acids are very important. Actually, they are coming from skeletal muscle when gluconeogenesis is going on. The easiest is alanine, which can right away to be transaminated to pyruvate, but there are so-called gluconeogenic amino acids that also can be turned into the carbon skeleton can be turned into glucose. Now you see urea cycle right away. So when we are producing ammonia from the amino acids that are coming into the liver for gluconeogenesis, this ammonia actually will join the urea cycle and produce urea, which can be eliminated, as I said before. So let's see with 2 cycles, which are mentioned in every biochemistry group book and also in your study guide for step 1. This is what we call the Cori cycle and the alanine cycle. Majorly, this is just a kind of an artificial thing to think about it, that some cells produce something that can be used by the liver to produce glucose, and when glucose is produced and it is secreted out to the blood, the same cell or any other cell can take it up and use it. So how does it work in the Cori cycle? The Cori cycle, the starting material is lactate. Now, I am not very happy about it that the drawer of this figure has chosen the muscle tissue and lots of red muscle cells, because optimally they don't produce lactate. They could, but they don't. So, but there are a lot of lactate producing anaerobic cells that will continuously pump out lactate. This lactate goes to liver, turns to glucose, glucose is pumped out, and glucose can be used by other cells. Now, why I am upset about muscle again? Because muscles do not use this glucose. They use fatty acids for energy. When the blood glucose is going under normal, they spare glucose for other tissues. So I would be more happy if a red blood cell would be depicted here. But this is just detail. So peripheral tissues producing lactate and utilizing the glucose which comes from that. Majorly, this is the Cori cycle. This is the alanine cycle. This is more complex, as you see. And yes, it is skeletal muscle that we need. So skeletal muscle is breaking down protein when a starvation signal, glucagon, epinephrine, mainly epinephrine for the muscle, hits the muscle and release amino acids. The main amino acid which is released is in the form of alanine, but there may be other amino acids too. So this alanine is going to the liver. This alanine is used to produce glucose. Now it is labeled here that when alanine goes to the liver, you have to deal with ammonia and produce urea. Glucose goes out to the circulation, and as I said, muscle is really not taking it back, but any other cell can take back the glucose. So what to remember? That alanine is coming from muscle. Lactate can come from any lactate-producing cells. When it is coming, when amino acids are used, ammonia needs to be removed, and that ATP is needed for both. Gluconeogenesis, to create something, to synthesize something, we require ATP, and we go there shortly. Pyruvate carboxylase is one of the enzymes that reverts pyruvate kinase. So pyruvate carboxylase will produce oxaloacetate, and then PEP carboxykinase will produce phosphoenol pyruvate. So going back here, this is pyruvate carboxylase. Actually, you see the pyruvate here, which produces oxaloacetate. And then in the, this is a mitochondrial enzyme, and oxaloacetate will turn to phosphoenol pyruvate. So this is PEP carboxykinase here in the cytosol, and then gluconeogenesis goes from there. Now, a lot of interesting things are happening in the mitochondria. So this is what I said. This is the enzyme, pyruvate kinase. This is the substrate. This is the end product. What is it before? This enzyme is working with a coenzyme, which we call biotin. Biotin is a vitamin like substance. And why do we need that? Because it will covalently link to the enzyme through a lysine residue. So this is pyruvate carboxylase. Why is it important? Because you see what the difference is between pyruvate and oxaloacetate. We have a carboxyl group. So biotin will help us to bind the carboxyl group out of bicarbonate or carbon in the form of carbon dioxide, and then will deliver with the help of this enzyme to pyruvate to form oxaloacetate. All right, first question. Pyruvate carboxylase is an enzyme that you learned about before. Where? Okay, here is the answer. It was a so-called anaplerotic enzyme in the tricarboxylic acid or Krebs cycle. Pyruvate is taking, oxaloacetate is taking acetyl coenzyme A in the Krebs cycle. And when Krebs cycle intermediates are used for other things, if you remember, then this oxaloacetate needs to be replenished. So from pyruvate, from PDH, we are producing acetyl coenzyme A, which oxaloacetate will take to the Krebs cycle. But from pyruvate, we also produce oxaloacetate, which will take then acetyl coenzyme A to the Krebs cycle. Now, here, we are not going in that way. We are using oxaloacetate to be a substrate for PEP carboxykinase. So oxaloacetate cannot cross the mitochondrial inner membrane. Oxaloacetate is reduced to malate. Malate can cross the inner membrane and oxidize back to oxaloacetate. Where did you see this enzyme, which we call malate dehydrogenase, before? Hope you are correct. This was part of the malate oxaloacetate, malate aspartate shuttle system. You remember? That was working in an opposite direction to put the reducing equivalent of NADH to malate and then transfer the malate into the mitochondria and release NADH so that it can go to the terminal oxidation. So a lot of use of pyruvate carboxylase and the shuttle system malate dehydrogenase for supporting energy generation and also for supporting gluconeogenesis. So this is in the mitochondria, liver mitochondria. Where does the ATP and NADH come from that we need here? So you see we need ATP for the pyruvate carboxylase enzyme to bind this carbon dioxide to the enzyme through biotin, and we need NADH to reduce oxaloacetate to malate. Did you guess? It is coming from beta oxidation of fatty acids. Beta oxidation is also going on in the mitochondria. We need to support the gluconeogenesis process with ATPs. Thus, fatty acid will come to the mitochondria of the liver cells and produces ATP and also produces a lot of NADH and FADH. Right, so let's go now to the cytosol. So we have oxaloacetate here. Oxaloacetate is turned into phosphoenol pyruvate with the PEP carboxykinase enzyme, and we gain back that carbon dioxide that we put into oxaloacetate before. This step uses GTP. You remember, if we count everything back to glucose, which is 6 carbon and this is 3, we need to calculate that 2 ATP were used by the PC enzyme and 2 GTP ATP equivalents will be used for PEP carboxykinase. So there is a condition which we call pyruvate carboxylase deficiency. And if you read through this case, and I don't want to do it right now, these are all very important signs that the baby is suffering from pyruvate carboxylase deficiency. Seizures, central nervous system hypotonia, muscle, psychomotor retardation, both, poor head control, muscle problem, lactic acidosis, elevated plasma levels of pyruvate and alanine, all typical of pyruvate carboxylase. Now, you can test other cells aerobic cells, whether there is pyruvate carboxylase there, because you remember this is also used for helping the tricarboxylic acid cycle. So even there is no gluconeogenesis going on there, you can see whether pyruvate carboxylase is expressed, or you can detect even the point mutation there. So in this case, fibroblast was tested and 1% of the normal was found, way too low to support the system. Let's go back to the liver. So we saw energy deficiency, which is a Krebs cycle problem. We saw hypoglycemia, which is a gluconeogenesis problem. We see pyruvate and alanine concentration increased because this enzyme is not working. We also see lipid accumulation because if this cannot work, then the Krebs cycle will not work very well either, and the acetyl coenzyme A will be pushed despite the hormonal signal that would not really allow fatty acid synthesis in this situation to produce fatty acid. And lactic acidosis, again, because pyruvate now is forced to be turned to lactate. Can something be done? You probably remember we discussed it at the Krebs cycle, that we can support the Krebs cycle with other intermediates, including a specific amino acid aspartate, which can be transaminated directly to oxaloacetate. However, this is a really not good therapy. It cannot be used for long-term care. It cannot be used, and it is very expensive. So unfortunately, PC deficient babies many of them end up with mental retardation and very serious problems, and many of them die. Now, I promise I will be shorter because we spent a lot of time on this area here, and you understand now why it was important. And now we just need to address the other two irreversible glycolytic enzymes. And because they are called kinases, they really phosphorylate. The reversal enzyme in gluconeogenesis will be a phosphatase. So number 3 is fructose 1,6-biphosphatase. Number 4 is glucose 6-phosphatase to reverse the action of PFK1 and hexokinase or glucokinase. We are in the liver. So this seems to be easy, correct? At least compared to the pyruvate kinase reversal. However, glucose 6-phosphatase is not in the cytosol. The previous steps are, and this is not. Glucose 6-phosphate, which is coming backwards in glycolysis, so coming to gluconeogenic pathway, needs to go to the ER. For that one, we need a translocase on the ER membrane, which will put through glucose 6-phosphate from the cytosol to the ER. And in the ER, we need the phosphatase there. So the glucose 6-phosphatase is actually bound to the inner membrane of the ER system. And then we need another 2 transporters. One is to transport glucose out of the ER to the cytosol, and the other is inorganic phosphate transported out from the ER to the cytosol. Now, this is a glucose transporter, correct? And this is specific to the ER, and we call it GLUT7. What is the next step? Now the glucose which is produced from gluconeogenesis is in the cytosol. It needs to go out to the general circulation, out of the liver cells, and we use GLUT2. Remember, GLUT2 is reversible, so GLUT2 can accept glucose, but it also can put out glucose to the circulation. So when you gather the special nature of liver and how it is really doing a lot of things for us, you can remember a couple of things from here. So liver is specific because it has GLUT2. Liver is also special because it can release glucose, having gluconeogenesis placed in the cell, and also that the glucose which comes from glycogenolysis also can be pumped out or put out to the circulation. Very good points to remember. And please note, and I go to the next slide here, that glucose 6-phosphate deficiency is well known. This is a genetic disorder, and it can be either the problem with the translocase or problem with the enzyme. Does not really matter which one is not working properly. We call it in the same disease because the outcomes is the same. We call it one Gherkes, and type A when the type B when the enzyme is not working well because of mutations. We call it a glycogen storage disease just because the glucose 6-phosphate, which is coming from either gluconeogenesis or glycogenolysis, needs to use this lost enzyme, which is, again, as I said, liver specific. So, and what happens when this enzyme is not working? Glycogen will accumulate in the cell and cannot be used by the liver to balance blood glucose. Probably I should have put here something with big red letters. Having gluconeogenesis in the liver, having this special enzyme and transport system in the liver, ER, glucose 6-phosphate coming from liver glycogen and gluconeogenesis makes sure that liver will be able to balance blood glucose. So when the blood glucose level drops, both of these processes will be upregulated in the liver. Both of them will help to put out enough glucose to the circulation to keep that blood glucose level in the normal range. Otherwise, you pass out, your cells cannot work well. You know what is happening in low circulating glucose conditions. All right. Now this will be even faster. We can synthesize glucose from other things. You remember amino acids. And at this point, I just wanted to remind you that we have glucogenic, ketogenic, and glucoketogenic amino acids. We can use the carbon skeleton of the glucogenic amino acids. We can use part of the skeleton of the glucoketogenic amino acids for glucose synthesis. We never can use leucine and lysine, which is purely ketogenic amino acid, for glucose synthesis. And please remember, when we start from amino acids, we remove the amino group. We have an ammonia load on the liver, so ammonia needs to be handled by the urea cycle. Why it is so important to have infants to get all this glucose? They have a larger brain to body weight ratio, which means that they will use more glucose. Frequent feeding with glucose-rich material is very important for the newborns and infants. They also have limited capacity for ketogenesis, so they cannot kind of replace partially, tiny little bit, glucose with ketone bodies. For newborns, the hepatic glucose synthetic machinery is not up to full speed yet. So it's very important to feed them with glucose. And the glycogen stores might not be even up to par, especially in premature and small for gestational age infants. Okay, glucose also can be synthesized from odd-chain fatty acids, as I mentioned before. So triglycerides will give you glycerol, which can go to glycerol phosphate and join the gluconeogenesis process. You remember we mentioned that glycerol can also be used for glycolysis. And these odd carbon fatty acids, I am repeating myself, propionyl coenzyme A, methylmalonyl coenzyme A, succinyl coenzyme A, goes to oxaloacetate. Please remember this enzyme needs vitamin B12. So in vitamin B12 deficiency, there will be a problem with channeling propionyl coenzyme A to gluconeogenesis. From other sugars, we can use fructose and galactose. Actually, mannose and galactose. Mannose we don't eat a lot, but if you eat mushrooms, there's mannose inside. It's that easy because these sugars need to be primed to sugar phosphates, and they can be turned into glucose 6-phosphate, which then can go to that last enzyme, glucose 6-phosphatase, and produce glucose. Fructose is a little bit more complicated. You remember that I talked about this fructokinase in glycolysis, that fructose, when it comes from high fructose corn syrup, fructose 1-phosphate will give the signal to the cell to accept more glucose. It's really not good for us. But then this fructose 1-phosphate, when we have fructose, can turn into gluconeogenesis. So you see here too, and this is an easier way to remember, that fructose 1-phosphate is 6 carbon. It needs to be split into 3 carbons, and it is a special enzyme, fructose 1-phosphate aldolase, which is similar to the aldolase which we use, but it is an isoenzyme which we use in glycolysis or gluconeogenesis. And then it can go into a funny way to join the gluconeogenic process. Now, how do we regulate this process? This is now a summary for you of what you learned before regulating glycolysis, and now compare and contrast with gluconeogenesis. We had the 3 irreversible enzymes, hexokinase/glucokinase, PFK1 and PK in glycolysis. You remember the allosteric regulators that you learned there. And then now this is gluconeogenesis, and you see that the opposing enzymes, the enzymes that circumvent PK and PFK1 and hexokinase glucokinase are listed there, and the allosteric regulation is listed there too. Now, what is circumventing PK? 2 enzymes, and out of which pyruvate carboxylase will be regulated by acetyl coenzyme A. This is again a cross-regulation by another metabolic pathway. And acetyl coenzyme A will be the positive regulator. Why? Because acetyl coenzyme A is produced in the mitochondria from fatty acid beta oxidation. You remember pyruvate carboxylase is in the mitochondria, and it senses that energy is available for gluconeogenesis. Does this acetyl coenzyme A upregulates the very first very important enzyme, and then gluconeogenesis can start. When we come to the biphosphatase that circumvent PFK1, you see cross-regulation between the 2 pathways, but this is opposite. Opposite pathways need to be regulated in an opposite way. So if you see AMP upregulating glycolysis, because lack of energy, AMP will halt gluconeogenesis. If there is no energy, gluconeogenesis cannot go, so it's better to stop it. Now you remember fructose 2,6-biphosphate, how important it was to push glycolysis ahead. But when glycolysis is pushed, gluconeogenesis will be stopped by the same regulator. So, couple of things to remember. Review glycolysis regulation and remember the different cross-regulatory agents that regulate gluconeogenesis. AMP, low cellular energy, acetyl coenzyme A, energy is available. How do we do the hormonal regulation? So this is a tiny little bit more complex than in glycolysis. Think about this one. Glucagon is a hormone which is released by pancreatic cells when they sense low circulating glucose level. So that must upregulate gluconeogenesis. What way? Again, I am building a story here, so I will look not only for gluconeogenesis, but for other pathways too. 1. Glucagon signal to the liver cell will upregulate beta oxidation of fatty acids. Good for us because it produces energy. It also produces acetyl coenzyme A, which will upregulate pyruvate carboxylase activity. Glucagon downregulates pyruvate kinase, which means that when gluconeogenesis is going on, pyruvate kinase is not opposing this effect. Glycolysis stops. Now, glucagon also decreases the synthesis of fructose 2,6-biphosphate, which will stop glycolysis but upregulate gluconeogenesis. Remember these two figures? Glucagon kinases phosphorylating the kinase subunit and stopping glycolysis, which means that gluconeogenesis can go. Insulin opposing phosphatases, removing the phosphate from the kinase subunit of PFK2, will produce fructose 2,6-biphosphate, upregulating glycolysis, inhibiting gluconeogenesis. There is another hormonal regulation by glucagon and, of course, opposed by insulin, and this is in the gene transcription level. Glucagon will upregulate the transcription of the genes that code for 3 of the gluconeogenic enzymes. In the meantime, downregulate the expression of the glycolytic enzyme. So there will be less glycolytic enzyme and more gluconeogenic enzyme in the cytosol to perform the job. It also influences aminotransferases. So when amino acids are coming into the liver, they can be handled. Everything can be channeled to alanine or oxaloacetate, so gluconeogenesis can take them as substrate. So finally, again, I have this figure which you can review what we talked about, and you can review the regulation also. Now, fasting state means high glucagon. So you see that what I talked about, glucagon is doing all this to promote gluconeogenesis. Thank you. Oh, I'm sorry. Gluconeogenesis is an absolute necessity. Liver is the main organ. Kidney can help a little bit in starvation. It can start from lactate, amino acid, propionyl coenzyme A, and other sugars also can join the gluconeogenic process. 3 kinases in the glycolysis circumvented by 4 enzymes. One of them is, out of the 4, is mitochondrial. Please know the details. Glucagon is the main hormone that will start gluconeogenesis, sends the signal. And I mentioned shortly pyruvate carboxylase deficiency, which is actually a pretty good case to review both Krebs cycle and gluconeogenesis. Thank you.